All right, I am re-recording the prokaryotic lecture because of the time delay problems with the audio. So again, this is prokaryotes, and in this lecture I am discussing what we know about bacteria. Now archaea, of course, are prokaryotes also. We don't know as much about archaea because they're such unusual organisms, live in such unusual places. It's difficult to study them. However, we assume that many of the things that I tell you about bacteria is true for archaea as well. So what I'm saying is we're going to assume that what I'm saying is true for both bacteria and archaea. If I know it to be different, I will bring it to your attention. Otherwise, we assume it's true for all prokaryotes. All right. Shapes. There are three different basic shapes for bacteria. If a cell is perfectly round, then we call it a cocci. Cocci is plural, coccus is singular. If it's a rod-shaped organism, then we call it a bacillus, is singular, bacilli is plural. And if they're spiral-shaped, there's various different versions here. They can be complete helices, like a helical shape, or little curved comma-shaped organism, or twisted rod. We'll see some examples of these in just a moment. So here's some examples of cocci. Now, the shape of the bacteria would either be a coccus, bacillus, or spiral of some sort. The next thing that you need to consider when you're thinking about bacteria is not just the shape, but next is the arrangement. And the arrangement is how those cells are put together in three-dimensional space. For example, some organisms are a single coccus. They live by themselves, not connected to any other cell. But most of them do live in some association. Either they are diplococci, two cells together, or they are streptococci. Strepto is chain, a long chain, looks like beads on a string. Or tetrads. Tetrads are packets of 4 cells together. They're all in the same plane. Or they are sarcinae. Sarcinae are packets of 8 cells together. Or they are staphylococci, and staphylo means irregular clusters. I always think they look like grapes, these irregular clusters. Likewise, there are arrangement terms that we use for bacilli. The vast majority of bacilli exist as single, just one cell by themselves. Most of them live in that type of lifestyle. The other options, though, there are some, very few, that are diplobacilli, where you have 2 connected together. One of the really cool things to see in biology is a streptobacillus. A streptobacillus is this long chain of bacilli all connected together. Here's a picture of a really long streptobacillus, where here's the end of the chain. It goes all the way down and around, comes back and twists and folds on itself, all the way back through here, up and around, and eventually ends here. That's one big, long streptobacillus, all the bacilli connected together in one long chain. A couple of organisms, a couple of bacilli have that arrangement. And then at the bottom here, this is what we would call a coccobacillus or a cocobacillus. These are organisms that are kind of short and fat rods. Sometimes students will look at them and they say, well, they look more like an oval. I can't tell if it's a bacillus or if it's a cocci. It could be that it's a coccobacillus, this short fat rod sort of between cocci and bacillus. For the spiral terms, these spiral-shaped organisms, if it is a curved rod, looks like a comma, then we call it a vibrio. Vibrio is this curved, comma-shaped organism. A spirillum is an organism that has 2 or more curves or twists. So if you look at the structure here, 2 or more curves or twists, we call it a spirillum. A spirochete. A spirochete is different. It is highly flexible, very flexible structure. It looks sort of like a spring under the microscope. It's very, very twisted and curved like that. We're going to see that spirochetes have a structure called an axial filament, A-X-I-A-L filament. Right? Spirochetes have this axial filament, and that's what actually gives them this twisted structure. And we'll see it also causes spirochetes to spin through the media as they swim. So they kind of corkscrew and twist through the media as they swim. That's a spirochete, this highly flexible spring-like structure. Now this diagram is here to show some variety and the variety that exists inside of these terms. So for example, here's different kinds of cocci, here's several different kinds of bacilli. Notice some of them are long and thin, some of them are short and fat, some of them are kind of like a dog bone, thin in the center, fat on the ends, but they're all bacilli of some sort. And you can see some of the arrangements here as well. Here's tetrads and strepto and staphylo and sarcinae, various examples. Here are the vibrio, the curved rods. Here are the sporillum, right, those 2 or more curves. And here are the highly flexible and twisted spirochetes. Now while we're here, I want to bring up a topic that sometimes makes life difficult, especially for a new student in microbiology. At the bottom here, this term pleomorphism. Some species exhibit pleomorphism, and pleomorphism means variety in shape and size, even though they're the same species. So sometimes you'll be looking at an organism underneath the scope, and you're looking at it, and it looks sometimes like it's long and sometimes like it's short, and you can't quite tell what's going on. It looks like there's natural variety inside of that organism. Some species have that ability, and we call it pleomorphic, and that's a quality of some organisms. And I know it makes life difficult for students, but some of them are pleomorphic like that. All right. We're going to go through the structure of the prokaryotic cell. No perfect place to start here, but we're just going to start from the outside and work our way in. So, if you look at the prokaryotic cell, some things you already know about it, of course, no organelles, but it does have ribosomes. You know that it has peptidoglycan in its cell wall. You know that it has a circular genome, no nuclear envelope around it. Right? We're going to go through it in more detail, but again, starting from the outside structure, and the outermost structure of a prokaryote is the flagella. So we'll start there. All right. The prokaryotic flagella. Now, I believe that in your notes, the notes that I posted online, I had this as eukaryotic, so make sure you fix that. That was a typo, so make sure you put in there that this is the prokaryotic flagella, so you don't get confused later. All right, the prokaryotic flagella here. A couple of interesting things. There are actually 3 parts to the prokaryotic flagella. The filament is the long piece that extends off the surface. That's what people think of when they think of flagella. They're actually thinking about the filament portion of the flagella. Then down in the cell wall, you have what's called the basal body. The basal body is this securing mechanism that holds the flagella into the cell wall and cell membrane. If you look at the structure of the basal body, you'll see individual rings, and then this piston structure that goes down the center. And that's going to actually, the piston will spin inside of the rings, allowing this flagella to completely rotate. The hook is what holds the filament onto the basal body. So again, you have the filament is the long piece, the hook holds the filament onto the basal body, and the basal body is this ring and piston structure that secures the flagella into the cell wall. Now, all portions of this flagella are made of a protein called flagellin. Right? This is the name of the protein that makes up the prokaryotic flagella. That's important because we're going to see that it's different substance makes the eukaryotic flagella. Now, because of the way the flagella is attached into the membrane with the basal body, the flagella does rotate 360 degrees. The prokaryotic flagella does rotate completely around, and it is that rotational movement that creates mobility for the cell. couple of other interesting things there. The flagella of a prokaryote does extend outside of the cell wall and the cell membrane. In fact, here you can see the membrane of the cell, right? Cytoplasm would be on the bottom here. Here would be cytoplasm. Here's the membrane of the cell, and the cell wall is outside of that membrane, and the flagella truly sticks off the surface of the cell. It is extending outside of the cell wall and the cell membrane. Now, as for what cells have flagella and what prokaryotic cells do not have flagella, I can give you a couple of guidelines. All of the spirilla have a flagella. They're all motile via flagella, so all spirilla have a flagella. About half of the bacilli have a flagella and are motile. The other half aren't. They don't have a flagella. But very, very few of the cocci have a flagella. Most of the cocci are non-motile. They don't have flagella. Now we can describe organisms according to their flagellar arrangements. So you could call a cell monotrichous if it had one flagella extending from the pole of the cell. We would say it was a monotrichous organism. Some organisms have two flagella, one at each end. Right? That's called amphitrichous. Amphitrichous are organisms that have two flagella, one at each end. Lophotrichous. Lophotrichous is when the bacteria has a cluster or bundle of flagella and they're all located at one end of the cell. Okay? That's lophotrichous. See that cluster all at one end? Here's peritrichous. Peritrichous is when the cell has flagella all over the surface. So flagella are coming out from all sides. That's peritrichous. So I would expect that if I described an organism as being amphitrichous, you would know that that means it has two flagella, one at each end. Or if I showed you a picture like this one and I asked you which of these appropriately describes it, you would know that this is a monotrichous organism with just one flagella. Now, regardless of whether the cell has one flagella, it's monotrichous, or if it has lots of flagella, the flagella are all used for motility. That's the job of the flagella. And remember, the prokaryotic flagella is rotated 360 degrees to push the cell through the media. It turns out that when the flagella is rotated counterclockwise, that pushes the cell forward. So that's what's happening here. When the flagella is rotated counterclockwise, that pushes the cell forward through the media. That's true even if it is a peritrichous organism with lots of flagella. When the organism rotates each of those flagella, and they each rotate on their own, as those flagella are rotated counterclockwise, they all bundle together in one bundle and they push the cell forward. When he rotates it clockwise, the flagella fall apart. So the cell can go through motility, can have motility by rotating their flagella in a counterclockwise rotation. This allows a function that we call chemotaxis. Chemotaxis is movement in response to chemical stimuli. So this is movement in response to chemicals of some sort. Now, chemotaxis, what kinds of things would a bacteria want to respond to in its environment? If the bacteria is in its environment, what sorts of things does it want to be able to move towards? What acts as an attractant? For bacteria, attractants are commonly nutrient sources or vitamin sources. They'll swim towards nutrients. Bacteria will also swim towards oxygen. Photosynthetic bacteria will swim towards light, right? Those are common attractants. If you think about things that would be repellents that the bacteria would swim away from, generally those are some sorts of toxins or waste products. They'll swim away from those toxins. So that's chemotaxis, movement in response to chemical stimuli. And they do chemotaxis simply by rotating their flagella. So here you see this peritrichous organism. It's rotating its flagella in a counterclockwise rotation, and that's pushing the cell forward towards something it's attracted to. If it senses there's a toxin in that environment, it will rotate its flagella clockwise the flagella fly apart and it says, oh, I don't think I like this environment very much. It reorients itself towards a good environment and rotates its flagella counterclockwise again to push the cell forward. So bacteria can alternate between rotating their flagella counterclockwise to push the cell forward or clockwise to stop the movement. And that's generally how they run chemotaxis, how they move in a positive direction. Now, while we're here, this term chemotaxis only means movement according to chemical concentration. It's not a bacterial specific term. In fact, yes, it's true, bacteria do chemotaxis, but so do other kinds of cells. For example, white blood cells do chemotaxis. They're capable of sensing things in their environment and moving according to that concentration. In fact, white blood cells will chemotax towards a pathogen. If they sense a pathogen in your body, your white blood cells will move towards it. So chemotaxis is not a specific term to bacteria. It just means movement in response to chemicals. While we're here, I want to bring up this structure we talked briefly about before. This is called the axial filament. The axial filament, this is simply a flagella, but it's a flagella that's not completely away from the cell. Let me say that more clearly. This is a flagella that's attached at one end. You see the flagella is attached here, and then it's wrapped around the entire cell. You can see in that picture they're showing you the cell in red, right, and they're showing you the flagella in yellow attached here and wrapped around the entire cell, and it's underneath what they call the outer sheath. So an axial filament is simply a flagella that's wrapped around the cell and then covered by the outer sheath. Now, as you look at this diagram, here you see a cross-section. Here's the cell itself. Here's the cell membrane in purple. Here's the peptidoglycan, this little hatched structure they're showing you, the cell wall. The axial filaments then are wrapped around. You can see they're showing several axial filaments here. The dots are wrapped around the cell outside of the membrane, outside of the cell wall, and then covered by this extra piece called the outer sheath. The only organisms that have axial filaments are called spirochetes. Remember, the spirochetes are those spring-like cells, highly flexible. They move by corkscrewing through the media. What gives them that unusual shape and movement is the axial filament. Because their flagella are wrapped around them, as they rotate their flagella counterclockwise to move the cell, as they rotate their flagella, they're actually putting pressure on the shape of the cell, and the cell is constantly changing shape and corkscrewing as it moves through the media, and that they're highly flexible in that way. Only organisms that have this are a spirochete. Now, the axial filament is made of the same stuff that a flagella is. In fact, it is a flagella. It's just wrapped around the spirochete and covered by the outer sheath. So it's made of flagellin. It has the hook and the basal body and all that stuff as well. Another structure that sticks off the surface of a bacterial cell. These are fimbriae. Fimbriae are short, fine sort of hairs that stick off the surface of some bacterial cells. They are actually made of flagellin as well, so they're sort of like short flagella, but there are many of them on one cell. At the very, very tips, however, at the very tips of each one of those hairs are carbohydrates, and so when I say carbohydrates, you should think sticky. So, for example, something that has carbohydrates or sugars, if you were to spill soda on your kitchen floor, you would think about sticky. And indeed, that's what carbohydrates do. Carbohydrates or sugars create a real sticky sensation. So, indeed, the fimbriae are used mainly for adhesion and attachment. They're very, very sticky. It allows the cells to stick to one another. The fimbriae allowed the cells to stick to a solid surface. The fimbriae allowed the cells to stick to our cells during an infection. So if you look at this figure, for example, they're looking at an organism. This is E. coli. E. coli is attaching to the intestinal cells via the fimbriae. Fimbriae allow attachment. In fact, if we look at an organism like Gonococcus is the causative agent for gonorrhea. It is a diplococci, and in fact, it has fimbriae. Gonococcus has fimbriae. And the fimbriae attach to the epithelial cells of mucosal membranes during an infection. Well, it turns out that if you take Gonococcus and you strip off its fimbriae, it's no longer infectious. Apparently, the fimbriae are absolutely required for Gonococcus to go through its infection cycle. So fimbriae actually do make a cell more pathogenic than a cell that doesn't have fimbriae. All right, another structure that's off the surface of a prokaryotic cell, some prokaryotic cells, and this is pili. Pili is plural. Pilus is singular. Sometimes it's called sex pilus or sex pili because it's used for a type of sexual reproduction. Pili are rigid tubular structures. One bacteria will build this tube structure to contact a neighboring cell. And what they do is they will send a copy of their DNA through the pilus to the other cell. That's called conjugation. So conjugation is a mating process with the transfer of DNA from one cell to another through the pilus. So conjugation is a mating process transferring DNA from one cell to another through a pilus. Now, conjugation, although it's extremely useful because it shares DNA between cells, it's a very, very uncommon event. Very few cells can even do conjugation. It's only a group of bacteria called the gram-negatives that can even do conjugation. And even then, the gram-negatives will only do conjugation with close relatives. They'll only go through that process with organisms they are closely related to. And even then, it's only in a really good environment. It's only when things are perfectly right that they will go through conjugation. So it's really a very rare event. But possible. It does allow bacteria to share DNA between individuals. All right. We talked about structures that stick off the surface of a bacterial cell. I want to talk now about the actual cell envelope. The cell envelope, what's surrounding the cell and separating it from the outside world. The cell envelope of a prokaryotic cell really has three different structures to it, three different layers. Each of those layers is made of something different, a different substance. And together they make a very strong protective coat against the outside world. So if you look here at the cell envelope, on the innermost is the membrane of the cell. Outside of the membrane is the cell wall, protection coat. And on the outside of that is called the glycocalyx, and it's made of carbs, carbohydrates. So the three together form what we call the cell envelope. So this term cell envelope encompasses the membrane, the wall, and the glycocalyx all together, each with a specific function, and together they work as a protection unit. I always think about this like a coconut. If you think about the coconut, on the innermost is the white meaty part, and then you have a thin but tough and protective brown portion, and then on the outside you have those hairy bits, and each one is different in its structure and function, but together they make a strong protective coat against the outside world, and that's the same way for the cell envelope. So again, we're going to go from the outside and work our way in. So the outermost portion of the cell envelope is the glycocalyx, so we're going to talk about that next. The glycocalyx, outside of the cell wall, it's the outermost portion of the envelope. The glycocalyx is made of carbohydrates. There are some proteins in there as well, but its main structure is made of carbohydrates. Now, I say carbohydrates, and you should be thinking sticky. Carbohydrates or sugars are sticky, and indeed that's a big function of the glycocalyx is attachment. The glycocalyx provides attachment for cells to attach to one another, for cells to attach to solid surfaces, for cells to attach to our tissues during an infection. The glycocalyx also provides protection, and mainly what I mean is protection against water loss. Carbohydrates will hold fluid in their structure. They retain some fluid, and so that kind of creates this moist environment around the cell. So the glycocalyx is holding water and moisture in and around the cell so the cell doesn't dry out. The other thing that the glycocalyx does is it helps bacterial cells to avoid phagocytes, and I'm talking about your white blood cells. Your white blood cells are looking for bacteria. When they find bacteria in your body, they're going to ingest them and destroy them. That's phagocytosis. The glycocalyx protects the bacteria against your white blood cells. Turns out that your white blood cells cannot kill a bacteria that has a significantly large glycocalyx. Now, there are a couple of different kinds of glycocalyces. Two different types here, what's called a slime layer you're seeing on top versus a capsule you're seeing on the bottom. Now, really it's the same thing. Both the slime layer and the capsule are made up of carbohydrates mainly and some proteins. It's exactly the same thing. The difference is how neatly those carbs are arranged. A slime layer is a loose, not very well organized structure. The carbs are just sort of grouped on top, while the capsule is a neatly organized structure, but really, potato, it's the same thing. They're all made of carbohydrates and proteins. It just has to do with how tightly packed those carbs are, as to if they call it a slime layer or a capsule. So if, for example, you had a whole bunch of Legos, right? If you had a whole bunch of Legos, there's two options really. You could either just group all those Legos up together in a great big pile, and that would be called like a slime layer, or you could very carefully click all the Legos into place and build a really nice, neat structure. That would be the capsule. They're still Legos, whether they're piled up or snapped together, and the same is true for the glycocalyx. It's still carbs, one way or the other. It's just how neatly organized they are. Now, how important is the glycocalyx? Turns out that the glycocalyx is really important for many bacterial cells. For example, an organism called Streptococcus pneumoniae. Streptococcus pneumoniae, as you can imagine, causes pneumonia. It also can cause ear infections and various other infections, even meningitis in some cases. Well, it turns out Strep pneumoniae has a huge capsule. In fact, I have a picture here. This is Strep pneumoniae growing on the surface of an agar plate. And Strep pneumoniae has a huge, big capsule around it. Remember, the capsule is allowing attachment, the capsule is retaining liquids, moisture, and the capsule is also protecting against white blood cells. Here you're seeing Strep pneumoniae growing on the surface of a plate, and notice how these colonies look big and thick and wet and moist. We say that these are mucoid colonies because they look like mucus or snot. Moist colonies. Well, Strep pneumoniae, apparently that capsule is vital for its ability to infect. If you rip the capsule off of Strep pneumoniae, these are the sorts of colonies you see. Here you're seeing rough colonies, non-mucoid. It's the same exact organism as up here. It's just removed its capsule. Notice how they look dried out and shriveled. They don't look moist the way they did before. Well, it turns out this Strep pneumoniae without his capsule is not pathogenic. He can't cause an infection. So apparently the glycocalyx, in this case a capsule, is vital for Strep pneumoniae's ability to infect. Here's a capsule stain where they're showing the capsule. Now, a capsule stain, by the way, is a negative stain where the background is stained and then the organisms stay clear. But then on top of that, they add a secondary dye. So that's why it looks kind of speckled in appearance. So, for example, here's a cell. There's Strep pneumoniae in the center. Here's the background. That huge clear area that looks orange in this picture, that huge big clear area is the capsule. Notice that the capsule is what? 20, maybe 50 times larger than the cell itself. That's a huge, thick layer of carbohydrates surrounding the cell, protecting the cell against water loss and against your white blood cells. So the capsule can be quite large. Here's another picture of a glycocalyx here. This is Staphylococcus, round cells in irregular clusters, and it's attaching to the surface of a catheter. This is an indwelling catheter, I believe. And catheter has smooth plastic tips, so we don't think about organisms being able to attach to it, but in fact they can if they have a glycocalyx. In this case, it's a slime layer that staph is producing, and that slime layer is allowing it to attach to the surface of the catheter through that structure. And that brings us to an important topic currently in microbiology called biofilms. A biofilm is a mix of microbes living together on a solid surface. A biofilm is a mix of microbes living together on a solid surface. Well, how is that, sorry, let me back up. So what does that mean? When we think about ecology, we tend to think about large environments. So for example, if you think about something like the rainforest and how the organisms interact and depend on one another, in the rainforest you have great big trees, and the trees are producing fruits, and the monkeys are eating the fruit, and then there's another organism that's eating the fruits, like a bird, and then there's epiphytes that are living up in the trees, and everybody's depending on one another, right? That's the kind of ecology we're used to. But in fact, that happens on a microbial scale as well. Here in this biofilm, you have a mix of different microbial species all living together, depending on one another, and they're attached to a solid surface. So maybe the brown microbe is making a waste product that the red microbe is ingesting, and maybe the red microbe is producing a vitamin that the green microbe needs, and maybe the green one is making a waste product that the yellow one eats, and that's the way it works. They're all living together in a relationship together, depending on one another, and they're attached to a solid surface. We would call it a biofilm. Well, how does that biofilm form? How does this biofilm become attached to a solid surface? This all starts because the very first microbes the first colonists have a glycocalyx. And that glycocalyx allows them to attach to the solid surface. And then eventually you have so many of them attached to the solid surface that their glycocalyx becomes a great place for other microbes to attach. So biofilms form because the very first colonists had a glycocalyx, allowing them to attach to the solid surface, and everybody else comes in and attaches to that glycocalyx of the original organisms. It turns out that as we look for biofilms, we can find them all over the place. Places where you don't think about microbes growing, we'll find biofilms there. So for example, you have a biofilm currently growing on your teeth. We call it plaque. Plaque is a biofilm, several different microbial species living together on a solid surface. You go into the dental hygienist and she scrapes that plaque off your teeth, and within a couple of hours it's already coming back. You already have biofilm reforming. We see biofilm growing on knee replacements. So we see biofilms growing on indwelling catheters. We see biofilms growing basically everywhere we look for them on a solid surface. You find them if you know what you're looking for. I did some work on biofilms that were growing on the inside of stainless steel pipes that were used to transport water. So they are truly everywhere, and we're only beginning to understand how they function. All right. So if we look back at the cell envelope here, we have talked about the outermost structure, which is the glycocalyx, that sugary coat on the outside that allows attachment and protection. Next thing I want to talk about is the cell wall. The cell wall is the middle structure of the cell envelope. The main job of the cell wall is to provide a rigid structure to hold the shape and the integrity of the cell. It prevents osmotic lysis. i.e. bursting, right? It prevents the cell from bursting open. In a bacteria, the cell wall is composed of peptidoglycan. And we're going to talk more about that in just a moment. Almost all prokaryotes have a cell wall of some sort, and in bacteria, it's almost always peptidoglycan. So what is peptidoglycan? Let's look at this structure of what makes peptidoglycan. Peptidoglycan is a polymer of glycans, which are a type of sugar, a type of carbohydrate. Two different glycans together and alternating in these strings. So here we have N-acetylglucosamine, or NAG, and N-acetylmuramic acid, or NAM. And if you look, each individual string is alternating NAG, NAM, NAG, NAM. Right? And you have multiple strings of NAM, NAG, NAM, NAG, NAM. Here you have NAM, NAG, NAM, NAG, etc. You have these strings of alternating NAG, NAM, NAG, NAM, NAG, NAM, and those individual strings are linked together by proteins. Proteins are linking the NAMs together. See that? All together, this entire structure is called peptidoglycan. So peptidoglycan is the alternating NAG-NAM strings attached together through proteins between the NAM groups. Okay? That's what peptidoglycan is. So from now on, whenever I say the term peptidoglycan, this is what you should be thinking of, these alternating NAG-NAMs attached by proteins. Altogether, this structure is very strong yet flexible, so it makes a really great cell wall material because it's strong and flexible, protecting against the outside world. Now, peptidoglycan, I can't stress this enough, peptidoglycan is only found in bacteria. Highlights, underline. Peptidoglycan is only found in bacteria. There is no peptidoglycan in any eukaryote. There is no peptidoglycan in archaea. There is only peptidoglycan in bacteria. This makes peptidoglycan a great target for antimicrobial treatment. If you wanted to kill bacterial cells without hurting human cells, you could treat your patient with some drug that kills and destroys peptidoglycan. If you destroy the peptidoglycan, you'll kill off the bacterial cell without hurting the human cell. Right? So it makes a really good target for antimicrobial treatments. And in fact, many common antibiotics do exactly that. Oh, excuse me, yawning. Many current antibiotics, such as penicillin and amoxicillin, they work by destroying peptidoglycan so that they kill the bacteria without hurting the human cell. Now, in bacterial cells, there are two very common cell wall structures. They're what we call gram-positive and gram-negative. We're going to go through each. We'll look at the gram-positive cell wall structure first. In a gram-positive cell wall structure, here you see the gram-positive cell. They've kind of peeled up a piece of the envelope so that you can see what's going on, a cross-section. In a gram-positive cell wall structure, you have the cell membrane here at the bottom. Okay. And then you have a thick layer of peptidoglycan. They're showing in brown. This thick layer of peptidoglycan. Now remember, if you could zero in on that peptidoglycan, what you would see is NAG-NAM, NAG-NAM, NAG-NAM connected through protein cross-links. And so you have this thick layer of peptidoglycan. In fact, it's incredibly thick, somewhere between 20 to 80 nanometers thick, which is huge compared to the cell wall size itself. That peptidoglycan is attached to the membrane through what are called teichoic acids. Teichoic acids attach the peptidoglycan down to the membrane so that it all stays together. This is called a gram-positive cell wall structure. So if somebody says an organism is gram-positive, it means that this is the cell wall structure. It has thick peptidoglycan, teichoic acids holding the peptidoglycan onto the cell membrane. This is different than a gram-negative cell wall structure. A gram-negative cell wall structure has two membranes, an inner membrane and an outer membrane. You see that here. Here's the inner membrane. Here's the outer membrane. The space between the two membranes is called the periplasm, or the periplasmic space. In that periplasm, there is a thin layer of peptidoglycan that they're showing in brown here. Thin layer of peptidoglycan running down the periplasm. There are no teichoic acids. Notice, no teichoic acids in this structure. There is something additional here to a gram-negative. A gram-negative does have what we call lipopolysaccharides, or LPS, located in the outer membrane. They're showing them in green in this picture. So it does have these lipopolysaccharides in the outer membrane. So a gram-negative compared to a gram-positive. The gram-negative has two membranes. Space between the membranes is the periplasm. Thin layer of peptidoglycan going down the center of the periplasm. No teichoic acids, but it does have lipopolysaccharides in the outer membrane. And it turns out that the LPS will act as a toxin, so it is toxigenic to humans, the lipopolysaccharides. Now, the gram-negative, because it has this extra membrane, it tends to be a little bit tougher to kill sometimes than a gram-positive. That membrane acts as sort of an extra filtering system, filtering out drugs and chemicals that may try to get into the cell. So it's sort of an extra layer of protection. So gram-positive and gram-negative, you definitely need to know differences between the two cell wall structures. Here's another view to compare gram-positive versus gram-negative. On the left-hand side here, a gram-positive cell wall. In yellow is the membrane, right? One membrane in a gram-positive. In brown is this thick layer of peptidoglycan, very, very thick. You can't see them here, but indeed there would be teichoic acids that are attaching that peptidoglycan to the cell membrane. On the gram-negative here, you have two membranes. The inner membrane is shown in yellow. The outer membrane is shown in green. Space between the two is called the periplasm. There is a thin layer of peptidoglycan shown in brown that runs right down the center here of the periplasm. No teichoic acids, but there are LPS in the outer membrane, lipopolysaccharides in that outer membrane. Now, it is really, from a bacteriologist perspective, it becomes really important to be able to determine if your bacteria has a gram-positive cell wall structure or a gram-negative cell wall structure. In fact, how we identify bacteria, that's generally the very first thing we look at, is whether it has a gram-positive or gram-negative cell wall structure. And that brings us to the Gram stain. The Gram stain was developed by a guy named Hans Christian Gram, and he was trying to find a way to differentiate between human cells and bacterial cells. That was his goal. He was never successful in his goal. He never actually found a staining process that did that very well. But he did accidentally stumble upon a staining process that we use even today, and it becomes very important. This is the Gram stain, and it helps us to determine if an organism has a gram-positive cell wall structure or a gram-negative cell wall structure. So in comparison here, gram-positive and gram-negative, we're going to go through the steps of the Gram stain. Actually, we've done this in lab already, so hopefully this should be reviewed a couple times now. We're going to talk about the steps of the Gram stain. You should know all of the different steps, what each thing is doing, what would happen to both a gram-positive cell during that step, and what would happen to a gram-negative cell. All right, so here you're looking at the gram-positive and gram-negative cell. The first step of a Gram stain is to use crystal violet. Crystal violet acts as the primary stain. Crystal violet, it turns out, stains peptidoglycan. That's what it does. Well, both cells have peptidoglycan, right? Thick in the gram-positive, thin in the gram-negative, but both of them do have peptidoglycan. Therefore, both cells are purple at the end of the first step. Next, you add Gram's iodine. Gram's iodine acts as what we call a mordant. Mordants permanently attach stains to their substrate. So it's permanently attaching the crystal violet dye onto the substrate here, which is peptidoglycan. That's what's happening when you add iodine. Right. Now, so at the end of this step, everybody had peptidoglycan, therefore everybody's still purple, both the gram-positive and the gram-negative. Now, the next step, we're going to add alcohol. Alcohol acts as what we call a decolorizer. First off, alcohol on top of peptidoglycan. Peptidoglycan is that thick carbohydrate structure. Alcohol does not penetrate it, so nothing happens there. However, on a gram-negative, you have a membrane on the outside. Well, it turns out that alcohol will very effectively dissolve fats and lipids, and that's what a membrane is. So the alcohol will dissolve the outer membrane of a gram-negative. Well, nothing's holding that peptidoglycan down, so when you dissolve the outer membrane, you also pull away the peptidoglycan and all of the purple dye. So at the end of the alcohol step, gram-positives would still be purple, but gram-negatives would be clear because you stripped off that peptidoglycan along with your outer membrane. Difficult to see a clear organism on a clear background, so the next step is to add safranin, which is acting as our counter stain. Safranin is a pinkish red dye, and it stains everything. So if you put a pinkish red dye on top of the gram-positive that's dark purple, it still looks dark purple. But if you put pinkish red on top of a clear gram-neg, now it's going to look pinkish red. So at the end of a properly performed gram stain, you would expect gram-positives to be purple and gram-negatives to be pinkish red. Now, what do I expect you to know from this gram stain? I would expect you to know what each of these substances does during the gram stain, right? What's the counter stain, what's the mordant, what's the alcohol. If I stopped you at any of these steps, you should know what color you would expect the cells to be and why. And there are other ways I could ask the questions too. For example, if you forgot to add the alcohol, what color would all cells be? If you forgot to add the alcohol, all cells would be purple because you never washed away the outer membrane of the gram-neg. If you got to the end of the gram stain and everything was pink, right, even things that you expected to be gram-positive, what would have caused that? If everything was pink, you would think, well, maybe I forgot to add the iodine, right? Those sorts of questions could come from there as well. So in the gram stain, I do want you to know the steps, and I want you to understand how that differentiates between a gram-positive cell and a gram-negative cell. All right. Moving on, in that cell envelope, we talked about the outermost structure, the glycocalyx. We talked about the cell wall, the peptidoglycan, and we talked. Now I want to talk about the innermost portion of the cell envelope, the plasma membrane. Now, the plasma membrane of a prokaryote is almost identical to what you've seen with a eukaryote before. As you look here, you have a phospholipid bilayer. Remember, the phospholipids are amphipathic, which means they're hydrophilic on each side and hydrophobic in the center where their tails are here. Right? That creates a very effective layer that is separating the outside and the inside of the cell through those phospholipid bilayers. There are proteins that extend through the bilayer. The proteins are largely used for transport, moving things across the bilayer. There are also carbohydrates. The carbohydrates are almost exclusively on the outside layer of the cell membrane. And the carbohydrates are generally there for signaling and for identification of the cell. This is the same for both proks and euks. You have the phospholipid bilayer, creating the boundary. You have the proteins for transport across that bilayer, and you have the carbs for identification. Altogether, this creates a selectively permeable layer. And selectively permeable means it allows some things to pass but not others. Right? So that's the important part of the cell. The cell has to be able to control what's inside of it and what doesn't come inside of it. And it does that through the plasma membrane because it is selectively permeable. The transport activity, of course, of the membrane is run mainly by the proteins. Proteins are allowing transport of things across the surface of that membrane. Now, a couple of differences. So far, everything's been the same between procs and eucs. A couple of differences between them. There are no what we call sterols in a prokaryotic cell. We're going to see in eukaryotes there are these structures in the cell membrane called sterols. There are no sterols in prokaryotes. The only exception is mycoplasma. I put it here for completeness sake. Stay tuned. I'm going to talk more about mycoplasma in a moment, but he's the only prokaryote that has sterols in his membrane. The other difference between procs and eucs in their plasma membrane is the fact that prokaryotes have the ATP synthesis enzymes in their plasma membrane. Remember, for a eukaryote, electron transport chain and ATP synthesis happens in the mitochondria. Well, a prokaryote doesn't have a mitochondria, so where are those enzymes located? The ATP synthesis enzymes are located in the plasma membrane. So that's another difference between procs and eucs, the fact that these metabolic enzymes, these ATP production enzymes, are located in the plasma membrane. All right, so what's possible with cell walls and cell membranes for bacteria? I started talking about this, but I want to kind of have one slide where we put it all together. There are really 4 possibilities for a cell wall structure in bacteria. The 2 biggies we've already talked about. So bacterial cell walls can either be gram-positive or gram-negative. You should know what that means. Gram-positive, the one membrane, thick peptidoglycan, teichoic acids holding the peptidoglycan on. Gram-negative, 2 membranes, thin peptidoglycan, LPS but no teichoic acids. 99% of bacteria are either gram-positive or gram-negative in their structure. Those are the very, very common structures that we see. There are a couple of exceptions, however. The first exception I want to talk about is these bacteria that don't have a cell wall. They're called mycoplasma. Mycoplasma do not have peptidoglycan at all. No peptidoglycan in mycoplasma. So they don't have any cell wall structure. They're not gram-positive or gram-negative. They have no peptidoglycan whatsoever. Now, because of that, they do have some of those sterols in their membrane in order to provide a little bit of extra strength and rigidity to the membrane. Other bacteria don't need that because they have peptidoglycan, but mycoplasma does. So they're the one bacterial genus that lacks peptidoglycan and does have sterols in their membrane. The other odd genus of bacteria here is called Mycobacterium. Mycobacterium have peptidoglycan. In fact, they have a good amount of peptidoglycan. But in addition to that peptidoglycan, they have what we call mycolic acids. Mycolic acids are thick and waxy, and they're going to make this a different cell wall structure than what we see for other organisms. So the structure of Mycobacterium we call acid-fast. And acid-fast indicates that it has these mycolic acids in its peptidoglycan. Nobody else has that. Only Mycobacterium has those mycolic acids. So for bacteria, there are 4 options here in the cell wall. There's the gram-positive, the gram-negative, these weird mycoplasma that have no peptidoglycan, but they do have sterols. And there's Mycobacterium that have peptidoglycan plus mycolic acids. Now it turns out if you did put Mycobacterium through a gram stain, right, if you did put him in a gram stain, he would turn out purple. He would look gram-positive because of all that peptidoglycan that he has. It does keep the crystal violet. So if you put him in a gram stain, he would look gram-positive, but really he's not gram-positive. He has the mycolic acids. If you put mycoplasma through a gram stain, right, He has no peptidoglycan, so what color would he be if you did a gram stain on him? He would be pink. He would look gram negative because he doesn't have any peptidoglycan. So those are the 4 options for bacterial cell walls. What about archaea? There's a lot of variety in that domain, in archaeal domain. Some archaea have protein structures for their cell walls. Some have polysaccharide structures for their cell walls. Some do not have any cell wall at all. The one generality that I can give you about archaea is that there is never peptidoglycan. Peptidoglycan is bacteria only. Highlight, underline. All right, archaea is various different substances, but never peptidoglycan. All right, moving inside of the cell. We've talked about projections off the surface, the flagella, the fimbriae, the pilus. We talked about the cell glycocalyx and the cell wall, peptidoglycan, and the plasma membrane. Now I want to work inside of the prokaryotic cell. On the inside of the prokaryotic cell is this dense jelly-like solution that we call cytoplasm. If you looked at the substance that makes up cytoplasm, it's mostly water, but there are some nutrients dissolved in that jelly solution. You will see sugars and amino acids and salts and all of the things that the cell is going to need for its metabolic function. Structures that are inside the cytoplasm. Now, there are no organelles, of course, because this is a prokaryote, but there are some structures inside of that prokaryotic cell. For example, there's what we call the nucleoid. I'm going to talk more about this in a moment, but the nucleoid is the region where the DNA is kept. There are ribosomes, of course. Those are 70S ribosomes. And some bacteria will also have what we call inclusions or granules. These inclusions and granules are storage sites, allowing the bacteria to store excess substances of some sort. All right, let's look through these structures, starting off with the nucleoid. The nucleoid is the region of the cell where the genome is kept. Now, it is not a nucleus because a nucleus has a membrane around the outside of it, that nuclear envelope. That's not what we have in a prokaryote. Prokaryote has no membrane surrounding it, so it's not a nucleus, but it is called the nucleoid region. So in other words, the genome is not just sort of scattered all over the cell, it is collected in one location, and that one location would be called the nucleoid. If we look at the genome of a bacteria, we'll see 2 separate portions. The chromosome, which we've talked about before. The bacterial chromosome is circular. Right? And they carry their DNA in a ring, different than eukaryotes, which have linear segmented genomes. It is a double helix, but it's circular. And bacteria only carry 1 copy of their genome, a single copy of their genome. Eukaryotes carry multiple copies. That's another main difference between them. In addition to the chromosome, bacteria will often carry what we call plasmids. Plasmids are smaller circular pieces of DNA. I often will call them bonus DNA. That's just sort of my term. It's not really a biological term, but what do I mean by bonus DNA? Plasmids carry genes that are not essential under most conditions. Right, so plasmids carry genes that are not essential under most conditions. So they carry strange genes, genes that may be useful but you don't need on a day-to-day basis. For example, common genes for the bacteria to have on its plasmid, they may carry antibiotic resistance genes. So normally the bacteria doesn't need to worry about antibiotics, but if it happens to have an antibiotic show up in the media, if it has antibiotic resistance genes, that's a bonus. And those bonus genes are carried largely on plasmids, where they have these extra pieces of DNA. Another example, the ability to do odd forms of metabolism. Those genes are generally carried on plasmids. So for example, I told you about bacteria that could be used in bioremediation, where you can sprinkle them onto an oil spill and they digest up the oil. Well, that's actually not a normal food source for those bacteria. In fact, normally those bacteria live on other food sources, but they happen to have plasmids that have the genes allowing them to digest the oil. So they don't need it on a day-to-day basis. It's not on their chromosome, but if the oil shows up, it's a bonus to be able to digest it. And so that kind of odd metabolic gene is found on a plasmid also. So again, they tend to carry genes that are not essential under most conditions. It's a bonus to have that DNA. Turns out plasmids are easily transferred from bacteria to bacteria, so they can easily transfer them back and forth. In fact, if one bacteria has an antibiotic resistance plasmid, you can easily transfer it to other bacteria in the area, which is how how antibiotic resistance spreads so quickly between organisms, is because it's on the plasmid and it's easily transferred. The bacterial ribosome here, or I should say the prokaryotic ribosome. A ribosome, you know that the prokaryotic ribosome is 70S. Turns out that the ribosome structure itself is actually made of both protein as well as a type of RNA called ribosomal RNA, or rRNA. So the structure is both rRNA and protein together forming these ribosome structures. Of course, the prokaryotic ribosome does differ in size and density versus the eukaryotic ribosome. If we look at the structure here, ribosomes have two subunits, what they call the large subunit and the small subunit, because the large subunit is larger and the small subunit is smaller. In a prokaryote, the large subunit is a 50S subunit, and the small subunit is a 30S subunit. And together, they form the 70S ribosome. Now, I realize that this is not math that you're used to, that 50 plus 30 equals 70, and I know that that's a little strange. But what's happening is this is actually a density measurement. S stands for Svedberg units. It's a measurement of density. So it doesn't exactly add up perfectly the way you would expect it to. So what I'm telling you is, I'm sorry the math doesn't work, but that's the way it is. The large subunit is 50S, small subunit is 30S, and together they form the 70S ribosome that's then active. The job of the ribosome, of course, is protein synthesis. This is where proteins are produced, is here on the ribosome, and for that reason, all cells have ribosomes. All cells have them. They're not an organelle. All cells on the planet have to have a ribosome. The other internal structure that you see from time to time in some bacteria are called inclusions and granules. Why is it called an inclusion versus a granule? It depends on what's being stored and how much of it. Inclusions and granules are both intracellular storage sites, so it allows the bacteria to store excess substances of some sort, depending on what the cell is. It could be storing different things. So they vary in size, they vary in how many there are, they vary in what they're storing, just depends on what bacteria it is. Some examples here, this is a bacteria that's storing excess food sources. Just as we store glycogen, they're storing glycogen. That's what you're seeing here. These are inclusions of glycogen. Some bacteria will store gas, like aquatic bacteria will store gas, and they can release or absorb more gas in order to get to the right depth in the water for those aquatic bacteria. These are photosynthetic bacteria that have sulfur granules. Sulfur is used in photosynthesis, and so when they find a sulfur source, they'll store the excess sulfur in these little granules to use later. There are other granules called metachromatic granules that will store phosphate. Lots of different options, just depending on the cell and its particular lifestyle. Here's a cool type of granule. This is called a magnetosome. Magnetosomes store crystals of iron oxide inside of them. Some soil bacteria have these magnetosomes where they're storing iron oxide, and they use them like magnets. Now, you know, Earth has a magnetic field, right? It runs from pole to pole. Well, it turns out that these soil bacteria that have magnetosomes use the iron oxide in the magnetosomes like a little magnet, and they will align themselves with Earth's magnetic field. Why are they doing that? Why do they care to align themselves with Earth's magnetic field? The short answer is we don't know. It's a very cool capability, and in fact you can do some really funny experiments where you take a dish containing these bacteria in them and you put it under the microscope and you line magnets up on each side of the dish, and all the bacteria will line themselves up with that magnetic field of the magnets around their dish. Well, if you switch the poles, all the bacteria will turn around and switch the other way. So kind of amusing, but why are they doing that? The short answer is we don't know. No doubt it has something to do with their ability to find the right environment in the soil. Is it controlling the depth that they're in the soil? We don't know. Does it allow them to use some sort of bizarre energy source? We don't know. But we know that they are indeed sensing magnetic fields and aligning themselves with the magnetic fields using this type of granule called a magnetosome, where they're storing iron oxide. The last structure that I want to talk about that some bacteria have are called endospores. Now, an endospore is a resting dormant stage of the cell. It's a way to survive in bad environments. I want to stress here that bacterial endospores are only formed by a couple of different cells. Most bacteria cannot form endospores. The only ones that can are 3 gram-positive genera. So just a couple of gram-positives can do this. No gram-negatives, and there are lots of gram-positives that cannot. The 3 bacterial genera that form endospores are Clostridium, Bacillus, and Sporosarcina. So, for example, Bacillus anthracis that causes anthrax can form spores. Bacillus cereus that forms a type of food poisoning can form spores. Clostridium tetani that causes tetanus can form spores. So there's only a couple of them that do this. Those that are in the genus Clostridium, the genus Bacillus, or the genus Sporosarcina. All right, so again, the spore is this resting or dormant stage. As long as the cell is in a good media, it goes through this normal cycle that all bacteria go through. It's a vegetative life cycle where the cell divides and gives rise to new cells, and then those cells divide and give rise to new cells. And as long as the cell's in a good nutrient environment with plenty of liquid, etc., it's going to stay in this vegetative life cycle. Well, if the liquid dries up or the nutrients run low, Clostridium, Bacillus, and Sporosarcina can start this sporulation cycle where they form an endospore. And what they do is they wrap their DNA up in a thick protein coat, okay? And they form a spore. And this spore can survive an extended time period. In fact, they're incredibly resistant to heat and chemicals and light. And they can lie dormant for an incredibly long time. What will happen is eventually, if they get into better nutrients, they'll germinate and go back to the vegetative life cycle. Okay? So these 3 genera can alternate between vegetative life cycle if everything's good, or forming a spore if things are bad and waiting for the media to improve. And when it does, they'll go back to vegetative. So the spore here, the structure, a spore is simply a thick protein coat, huge, big protein coat called the spore coat, surrounding the DNA that's wrapped up in the center. Spores are completely dehydrated and therefore metabolically inactive. There's absolutely nothing going on inside of them. No metabolism, no cell division, nothing. It's just the DNA wrapped up in this thick spore coat. Spores are the hardiest thing we know of anything on planet Earth, the hardiest life form. Spores will survive boiling, they'll survive drying, they'll survive freezing, radiation, chemicals, unbelievable ability to survive in all sorts of different environments. Here you're seeing, I believe this is Clostridium tetani. It's a bacillus. You can see the bacillus here, and the spores forming on one end, giving it sort of a safety pin look. In a bad environment, it's beginning to form the spore here in the end of the cell. The rest of the cell will die away, and that spore will survive through boiling and heat and desiccation and radiation and UV light and all that stuff. The spore will survive. So unbelievable ability to survive. In fact, the food canning industry uses spores as its guideline for sterility. If you can get things hot enough, long enough, to kill off a spore, then you know nothing else has survived. You truly have reached a sterile environment. That's kind of the way that we measure whether or not we have achieved sterility, is whether or not we've gotten hot enough to kill spores. So how long can these spores survive? Short answer, we don't know how long they can survive. I have a couple of shocking statistics for you. For a long time, the oldest spore that they knew of that had survived, they had isolated it from a bee that had been fossilized, and that bee was estimated to be 25 million years old. They did carbon dating, and to date the bee at it being 25 million years old. They isolated a spore from its gut, right? They put it into rich media, and it germinated. 25 million years that spore survived and then germinated once it returned to fresh media. That's pretty impressive ability to survive for a long time period. More recently, they identified a spore. They got it from salt crystals. They isolated the spore from salt crystals. They then put that spore into rich media and it germinated. And those salt crystals were dated at 250 million years. So unbelievable ability to survive long time periods, even with this protein coat protecting their DNA. And as soon as the media returns to a good state, rich media, then they'll germinate. Right? Those are endospores. Again, most bacteria can't do it. It's just a couple of genera that can, and those genera happen to be gram-positive. All right, that brings us to the end of the prokaryotic lecture, and I will see you guys next time.